All right, this is our last section for our membranes, where we're gonna talk about bulk transport. So we are gonna move a lot of stuff all at once. We're going to, we can move stuff into the cell with endocytosis. Um, endo means inside, exocytosis. Exo means outside. We're good, so we can move stuff out of the cell. Okay, and then we're gonna talk about some ways that endocytosis can be specific, where we're gonna only move certain things and not other things. Okay, so there's three flavors of endocytosis, where we're gonna bring things into the cell. Okay, so first is phagocytosis. Phago means to eat. Then we're gonna talk about pinocytosis, which pino means to drink. And then we'll talk about this specific, this receptor mediated. This is how we're gonna get specific. All right. Let's do it. Okay, so phagocytosis. Phago, eat. Cyto, cell. So this is our cell eating. So what we're gonna do is we're gonna have our large cell that is going to encounter some kind of food particle. This could be a bacterial cell. It could be just a organic molecule. It could be any number of things. What we're going to do is we're going to take that plasma membrane and we're just going to pinch it off. And we're just going to keep folding that plasma membrane until eventually this little bacterium gets totally trapped. And then what we can do is we can take this, this bacterium that's encased in part of our plasma membrane. We can have another a little guy with digestive juices. We can actually have these two things fuse. So here's our little bacterium. And now we release all of those digestive juices and break that bacterium down. So here we can see all these little foldings of the plasma membrane as it's starting to engulf this bacterium. So this is a IRL version of the drawing over there. This is called an electron micrograph. And so we can see things that are really, really small scale. So 1 micrometer is really tiny. Another version of endocytosis is pinocytosis. So pino is drinking. Cyto again is still cell. Okay, so we're going to do the same thing where we have various solutes dissolved in our extracellular matrix. We're going to fold in that plasma membrane and we're just invaginating this more and more until boop, finally that is trapped. So the function of pinocytosis is thought to be sampling the solutes in the extracellular matrix. So like what kind of solutes are out there? So if you've ever gone to a brewery or a bar and ordered a flight of beers, they bring you these little tiny glasses that are like, 2 or 3 ounces of beer. But you get a whole bunch of different kinds and then you can taste and see which kinds you like. That's kind of what the cell is doing. It's just kind of sampling the options to determine if it wants to bring in a whole bunch of those at once. Okay, so that's pinocytosis, pretty easy, straightforward. In our receptor mediated endocytosis, this is how we're going to get specific for our different types of solutes. Okay, So once we can identify what's out there, then we're gonna say, aha, all right, well, we only want this thing and we don't want all the rest of the crap. Okay, so what we are gonna have is we're gonna have these proteins that are embedded in the membrane. And this three-dimensional shape of this protein is going to specifically fit whichever the target solute is. Okay, so if we have other solutes in here, this is a triangle, it's not going to fit into this part of the protein, so it's that protein's not going to pick up that solute. Okay, so this is how we're being selective about what solutes are actually going to come in. Okay, so we grab that. We also have all these other little proteins. We don't need to worry about that word. We'll just know that these are other types of proteins. So again, we're folding in that plasma membrane until we have this, our solutes, our target solutes are encased. These little proteins act as a barcode that's the same as your Amazon delivery driver will scan to say, determine where this needs to be going. So this is so that it gets delivered to the correct place in the cell. All right, so exocytosis is when we are trying to get things out of the cell. So exo, out, or external cytosis cell. Okay, we're taking out the trash. So let's say that we have brought in a bacterium. We've broken it down, and now we have all this left over. We have all this waste products. Okay, this is bad stuff. We don't want this in our cell anymore. So we are going to have this little plasma membrane organelle that I'll tell you about later when you're older. It's going to travel to the plasma membrane, which is the right, that barrier of the cell. I'm going to say this cell boundary. Okay. This plasma membrane of this little organelle is going to fuse with the plasma membrane of the cell, and then all this bad stuff, all these waste products gets dumped out into the external environment of the cell. This one, I think this one really just looks like a puking. This is cell puke. I'm a professional.